So in summary, we compared these 5 different methods against 3 different datasets, and there were clear winners in both resource usage as well as integration fidelity. Integration is a very important part of single-cell analysis, but it's also one of the biggest challenges. There's no perfect integration method, so we're going to be testing 5 different methods on 3 different datasets. Probably the most well-known is this Seurat CCA integration that a lot of people do. We're also going to be using the Seurat RPCA, which is one of their newer methods, the Harmony method. And so these first 3 are going to be in R, but then we're going to also do 2 Python libraries. One is going to be SCVI, which is a deep probabilistic model, and Scanorama. So, of course, other people have done this. For example, this Nature Methods paper, which benchmarked a bunch of different integration methods. I'm going to be doing things slightly different than they did, and I will also walk you briefly through the examples and some of the differences between them that I find important. So for my first dataset, it's going to be this reprogramming or dedifferentiation dataset, where they take mouse fibroblasts and reprogram them to iPSCs. So what makes this dataset so great is they collect samples every 12 hours over 18 days. So one of the challenges of integration is that integration in some cases erases actual biological differences between samples. So what's great about this is since it's every 12 hours, between each sample, we know there's going to be a subtle biological difference that we don't want to get rid of. So this is going to be a pretty challenging data set to integrate. For my other data set, just getting PBMCs from this recent Nature Medicine paper. And what I'm doing here to make it an actual challenging problem for integration, I'm just going to take healthy. What I'm doing is in all but one sample, I'm getting rid of the CD16 positive monocytes. So only one sample is going to have CD16 positive monocytes. So what we want to see in a successful integration is that we still have a distinct population of CD16 positive monocytes, even though they were only found in one sample. And then for the last data set, it's kind of an unconventional idea, but what I'm doing is integrating lung and muscle data together. They should have some similar populations like endothelial cells and some immune cells, but I just wanted to get an idea of how these different methods react to some highly different cell populations. We don't want any aberrant clustering together of distinct cell types. Alright, so starting off with Seurat's CCA method, I'm not going to go line by line in the code, but I will make these notebooks available on my GitHub if you want to see these examples. I'm just going to run through the code very briefly. So I just load all the files as a list. So I have my ingestion function, where I just add some metadata, do some basic filtering. In all these examples, I'm just doing very basic filtering and pre-processing just to keep it the same in every one of my examples. But I will point out some of the differences here. So for CCA, we need to find the variable features for each data object individually. And then I'm just returning this, applying it to my list of files. So I get a data list or a vector of Seurat objects. So we need to use the select integration features on this data vector. And then we do the find integration anchors, and then we pass that anchor set to integrate data. So this one's going to be the simplest to evaluate because even with 128 gigs of RAM, I wasn't able to integrate these 90,000 cells. So with just 90,000 cells, we ran out of memory, but that's not before it ran for like 5 or 6 hours or something. So a big waste of time, and then it didn't even finish. Obviously, that doesn't say that this might not work well with smaller data sets, but for large data sets, unless you have a whole lot of RAM and time, you should be avoiding CCA. So how did the Seurat RPCA work instead? So a lot of this is similar. Basically, the only difference here is after we get the features, we have to do scaling and get the PCAs. So just a small function for that and applying it to that data vector. And then this is the same, except with the important difference of putting RPCA here. Oops, I must have erased the time here. But this took around 44 minutes, and at the end, I'll show a RAM usage comparison between them as well. But like I mentioned briefly before, RPCA is one of their newer integration methods. It's supposed to be a little more conservative when shoving things together, so in theory, it's going to erase less of the biological variance that CCA might. So after integration, scaling and processing on the integrated data, and then I'm just converting this to day. Like I said, there's 18 different days. I'm just converting that into an integer so we can plot. And here, so when we plot the actual days that the cells come from, we don't see any clear progression. Day 20 for the iPSCs seem to be distributed kind of in an almost random fashion throughout this. There's no clear time progression. And since I'm starting out with this as the first one you see, you'll see what it should look like in some of these other methods. At least for this data set, RPCA did not work well. But anyways, let's go to Harmony now. So for Harmony, we're still using Seurat as the data handler or the object because most people like to use Seurat. So the beginning part's going to be pretty similar, except we don't need to find the features here. We still get this data vector, but then we merge it into one object. So we just need to use this merge for the first object and then all the other objects from that data vector. And then for Harmony on that single object, we need to find the features, scale, and the PCAs, and then we can actually run Harmony. So Harmony on this data set only took around 10 minutes. And then we need to run UMAP, and we need to specify the reduction is Harmony. So when you run Harmony, it adds this 40 feature by how many cells matrix to your object that's saved under this Harmony slot. Anyways, we're doing the same thing here. And compared to the last one, the RPCA, we do see some stratification by day, but it's still not quite what we expect. We have these two different islands here. They're not connected, and I'm not 100% sure what's going on here, but it's not really what we expect biologically, but it is an improvement from RPCA. It seems to preserve some of that biological variation between the samples, and it only took 10 minutes instead of 44. Okay, so let's go on to SCVI. And again, I'll make this notebook available on GitHub. But anyways, similarly, I'm getting a list of files and then making a similar pre-processing function as to what I did in R, processing the data through that function. And then I combine it into one AData object with scan, peek, and cat. And then I'm just doing very default SCVI model setup here. So I'm just passing the sample as the categorical keys and the total counts as the covariate keys. Of course, we can make this a little more complex with mitochondrial ribosomal counts, but again, I'm just keeping all the pre-processing and keeping things as default as possible. So when we train the model on these 90,000 cells again, it took around 22 minutes with default settings. You can increase the number of epochs and make it longer. Theoretically, it might be a little better, but the default was 22 minutes. And so one of the big differences, at least relative to Harmony and Scanorama, which I'll show next, is that SCVI, you actually get normalized expression values. So like Harmony just made that Harmony reduction with those 40 dimensions, which is great for clustering and UMAP projection, but you can't get the actual values of the genes. SCVI actually gives you that normalized expression as a new matrix. So RPCA from Seurat corrects the values as well. But again, we saw that it didn't do a good job, at least with this data set. And SCVI allows you to do differential expression and other stuff using these corrected normalized expression values. But anyways, let's see how it did with this data set. So here we see a much clearer progression, where down here we see day 0 cells, and then they get brighter or as the day increases. But anyways, so SCVI seemed to do a pretty good job, and it doesn't erase this biological variation that we see between these samples. So Scanorama, it's going to be similar, except in the pre-processing, we need to normalize, convert to log, and find variable genes for each individual file. But this step is, only one line of code. It's pretty simple. We just pass this data list to integrate scanp, and that modifies all the individual datasets within it, and then we can just concat them. And then we just need to get the integrated matrices from each of the datasets, and we're saving that to this slot here in AData. And then we can use that slot for that reduction to calculate the neighbors and then the UMAP. And then here, this looks pretty similar to what we saw with SCVI. We get a nice population of day 0 that slowly goes out. Even here, we get this nice, very distinct cluster of iPSCs. So this did at least as good as SCVI, if not a tiny bit better. But again, with the Scanorama, you don't get the actual normalized values. You only get this dimension reduction. And Scanorama, on this data set only took 3 minutes. So out of all the methods, this was actually the quickest. And then if we look at the actual RAM usage over time, again in that CCA Seurat, it got up to here and then decided that was too much memory and failed. I don't know how much it would have actually taken. RPCA on those 90,000 cells still took a lot of memory. I needed around 90 gigs of memory here. Harmony, which you can see here in purple, it ends right here, was the quickest and most memory efficient of the 3 R methods. It's pretty on par with SCVI. And then Scanorama, of course, took the least amount of memory and was the quickest. All right, so let's move on to that CD16 population removal dataset, where there's only one of these 12 samples that actually has the CD16 positive cells. So this is all going to be very similar to before, except the data are in a different format, so this first step is going to be a little different. But this is going to be the same all the way down to here. So we have, in this case, 61,000 cells. So 61,000 cells was successfully integrated with CCA. How long did it take? Sorry, I skipped. So 1.3 hours. So not very quick, but you can go eat lunch or something. It's not the end of the world. And then the only difference here is I had the metadata for the cell type already, so I'm just merging that in, and then I can plot it by that. So do we get... Let me just pop that out. So our CD14 cells are mostly in here, and this is dendritic cells. Don't really see where the CD16 positive population is, but we can zoom in on that. So if we just look at the monocytes here, we see that they're in the same cluster. It looks like the CD16 positive ones are mostly down here, but they're in the same cluster as the CD14 ones. So it didn't separate the two. But let's see how RPCA does here. So actually R crashed when I ran that initial CCA analysis on the dedifferentiation dataset, and it didn't save some of my work, but basically it's the same as the pre-processing on the CCA I just showed, and then it's going to be similar to what we did above for this dedifferentiation dataset. But anyways, how did the data look? So it's unfortunate here that the coloring between CD16 and CD14 is very similar, but here is this population of monocytes, and these again are the dendritic cells, but actually we get this little peninsula which corresponds to the CD16 positive cells here, and if you don't believe me, we can look at those on their own, and of course now we see that the CD16 positive cells actually segregate into their own cluster here. So RPCA seems to have done a better job than CCA on this dataset. So we have harmony here with the cluster of monocytes and dendritic cells. And again, the coloring in Seurat's a little weird here, but here's our population of CD16 cells. And when we zoom in, we see that they clearly differentiate into their own cluster. So harmony seems to have done a good job here. But again, you only get the harmony reduction, you don't get normalized values. So moving on back to SCVI, just the loading of the data is going to be slightly different than what we did before, but the rest is basically going to be the same, around 62,000 cells. In this case, it took 19 minutes. We get our big population of CD14 cells, dendritic cells, and then CD16 is in its own very distinct cluster here. So it at least passed this test. We can tell that it looks like the annotations are slightly different from the original paper than what we have here. It looks like a big mess of T cells with slightly different annotations. So I'm not going to go into this, but that does stand out here. And then Scanorama, the pre-processing and other stuff is similar to what I showed before, so I'm not going to go over it. But we get this population of CD16 positive cells. And actually, this population of T cells looks a little more distinct than SCVI. So it looks like the integration from Scanorama, in this case, looks a little better anecdotally without looking into it more to see what actually happened. But so it looks like the only one that really failed this was the Seurat CCA analysis, which is kind of the OG analysis method. And a lot of people still use it and might not know there's better alternatives. And then finally, what happens when we integrate two datasets from different tissues? I'm not sure this is the best test of integration fidelity, but I was honestly just curious and wanted to see what happened. So the rest is kind of the same. I think there were 16,000 cells, and in this case, CCA only took 3 minutes. So when you have a small data set, CCA is perfectly fine in terms of time. So here's our two data sets together. Green, I just labeled all the lung cells because they weren't annotated already. The cells from the muscle dataset were. So you see like B cells are put together, endothelial cells. These were macrophages, I'm pretty sure down here. But we do see there are distinct populations. So even CCA doesn't force together very distinct populations like some of these muscle cells. But I do worry a little bit, especially because from lung you get a lot of alveolar macrophages. And I don't really think macrophages from muscle should be clustered with alveolar macrophages. So I think those are aberrantly put together here, and they shouldn't be. So for that same data set, RPCA takes 34 seconds, and these data are more or less similar. But there's two important differences, I think, here, is that this appears to be the big endothelial cluster here, but it's segregated between lung endothelial cells and muscle endothelial cells. So we do expect that endothelial cells in these two different tissues are slightly different, and in CCA they were kind of forced together. So RPCA, again, similar, but seems to do a slightly better job in this weird juxtaposition of different tissues. When we look at macrophages, though, it's still kind of doing something weird. These weird small clusters of macrophages in a combination of alveolar macrophages and muscle macrophages. So it still seems a little off, but again, this was a weird combination of data sets. Harmony seems to have done a good job, at least aesthetically. This looks more pleasing. Clusters seem more distinct. We get this segregation of endothelial cells. We get these distinct muscle populations on their own. Still get a little bit of funky stuff with the macrophages. But let's see how SCVI does. So just off the bat, it does seem like there's less overlap between any of the clusters here, and I would hazard to say that's a good thing. This is our big alveolar macrophage population, and we see that the muscle macrophages segregate into their own groups, which is good. Endothelial cells from muscles versus endothelial cells from the lung. I do think I would have liked that these B cells from the muscle and the lung integrated a little better, but they are kind of in the same proximity. And then again, one of the benefits of SCVI, I just wanted to point out here, you get these normalized counts. For example, if we wanted to plot CDH5 endothelial marker, can we get those endothelial cells from both of the two data sets? And then CD45, this is just all the immune cells. And then finally, how did Scanorama do? So Scanorama, which had been our best performing method in most of those other tests, actually seems to have done the worst in this one. You get this weird, big conglomerate cluster that I'm not really sure what's going on. There's no distinct clustering that's apparent. And then when we actually look at the cell types, we get this weird clustering that just makes no sense at all. So in my opinion, SCVI did the best for this data set. So in summary, we compared these 5 different methods against 3 different data sets, and there were clear winners in both resource usage and time, as well as integration fidelity. So for this dedifferentiation data set, Scanorama appeared to do the best, SCVI being a close second, and then none of the 3 R methods worked as expected. For our CD16 positive monocyte removal test, I think either Scanorama or Harmony probably did the best, but Seurat CCA was the only one that really failed this test. For the integration of 2 different tissue samples, SCVI did the best, and others not so well. So if you forced me to pick a winner, I'd probably say Scanorama, followed closely second by SCVI. Harmony did pretty great too, but at least with these 3 different data sets, RPCA and CCA didn't do so hot. But with the important caveat here, only SCVI and the Seurat methods actually give you corrected counts that you can use in downstream analysis. But if they correct the counts poorly, your downstream analysis is going to be skewed as well. So Seurat methods might be better if you know your data sets are similar, but at least in this case, they didn't do so well. So if you need normalized counts and want to do differential expression, then I would say from these tests, SCVI is the clear winner. So the results from this Nature Method paper more or less reflected the same thing, although they tested a whole bunch of methods. They showed that Scanorama, in their case, was number 1. This ScanV is a method within SCVI-tools that I didn't test, but it also uses a reference, and that's supposed to make it even better. Maybe I can do that in an upcoming video. But we see that Harmony did very well as well. And then somewhere in this mess of 55 additional rows not shown, there's the 2 Seurat methods.